everyone with an interest in MASH, or more broadly, steatotic liver disease, surfs up. Season 5, Episode 12 of Surfing the MASH Tsunami starts now. Before we get into this episode, I have a set of comments to make. This is a sad week throughout the steatotic liver disease community, and tremendously sad at Surfing the MASH Tsunami. Our co-founder, former co-host, sometime surfer, and North Star colleague, Stephen Harrison, passed away suddenly Tuesday morning. I'll have a little more to say about this during the business report, and we are planning an episode to honor Stephen sometime in the next couple of weeks. But for now, suffice it to say that Stephen achieved his goal of leaving a big fat dent in steatotic liver disease, now matched only by the size of the hole his death leaves in the global steatotic liver disease community. And with that, we'll skip opening quotes, introductions, and all that, and simply start our episode, which looks at the 2020 and 2023 views of why clinical trials fail. Stephen knew a lot about this subject. Of course, with the Rezdiffra approval and some of the other promising phase 2B and 3 compounds in development, we may have overcome many of the hurdles around failure, but it still felt worthwhile to look back one more time. Ironically, the 2020 episode on this topic was the first time Jörn appeared on this podcast. Stephen and Louise were the other participants. After this episode, I'll be back with a question of the week and, in place of a normal business report, my usual comments about Ukraine and Israel, and then some thoughts about Stephen. So we have with us Will Alazawi, who we've not seen since sometime last year. Will, how are you today?
I'm very well, thanks. Good to be back.
And I want you to know, Will, this is the clearest audio and video we've ever had with you on this podcast. In fact, as soon as you popped up and I saw your face, I said, this is going to be better. We'll actually be able to hear him this time. It's a good thing.
That's good. Glad to hear it.
Me too. And then we have Sven Francque with us. Sven, how are you doing this evening?
I'm fine, thank you, Roger. I'm happy to be with you today.
Delighted to have you here. And then Jörn is back. Good evening, Jörn. And then we have Louise. Louise, how are you this evening?
I'm very well, thank you very much. Nice to see the guys on again.
Preparing to change hemispheres on us again, hey?
I am. Although I'd like to say I'm going warmer, but I'm not. It's very autumnal in Perth at the moment, so probably about the same.
Perth is lovely, so it should be good. All right, Groundbreakers. So one good thing, personal or professional, or if you wish, one of each that have happened in the past week. Brave one, go first.
Oh, I'll jump in. Liverpool are winning again, so I'm happy. And Crystal Palace absolutely nailed it yesterday with scoring 5 goals, or 6 because they did score an own goal, so they've never scored that many goals. So I can talk Premiership again.
So I'm not looking at the table, but does that mean they'll finish 13th, which is what they always do anyway?
Well, it means we've got a new manager playing attacking football. We're going up the table and we'll stave off relegation, so my husband will be very happy. And my eldest, and me.
This is a good thing. Okay, next.
My mind doesn't go much beyond construction at the moment, I'm afraid. I'm having a lot of building work done, so we're moving out. So I've got blueprints in my head and steel bars and bricks all over the place. But which is why I'm pretty pleased that at least the internet's going well.
So does this mean that the next time you're on with us, the construction will be done and we'll be back to the previous poor quality of reception, or?
It depends if we have to wait another year till I come back on again.
No, it won't be that long. Okay, Sven or Jörn, go ahead.
Yeah, I can go ahead because we still had, not last week, but the week before was still Easter holidays. And we were talking about construction. We went to Barcelona and went to visit the Sagrada, of course, to see how it evolves with my daughter. So we had a splendid time there. And I also, my eyes were catching in one of the glass-stained windows the name of one of my staff members. So I made a picture and then I showed it to her, and she was, overwhelmed with it and very happy and surprised that her name is figuring on one of the glass-stained windows of the Sagrada Familia. But she's from Spain originally, so it's special for her.
Fantastic.
That's very cool.
Yeah, I'm sitting here, speaking of constructions, trying to reconstruct my audio and speaker systems. And I hope I'm live now with you guys. And it was a fun week. A little cold, kind of a winter storm here. Cold weather moving in from the north. Otherwise, everything is fine.
I had a great weekend. As people who listen to this podcast know, I love to go to a town on the Jersey Shore called Cape May, which we go to frequently. But they have 2 major jazz festivals a year, and for whatever reason, we never caught either of them until this past weekend when we were in Cape May for the Spring Jazz Festival, and it was remarkable. Branford Marsalis was the featured performer. He played for 90 minutes to a packed house on Saturday night, and he was brilliant. But what I liked even better was a lot of the younger entertainers that you don't really know. A woman in her 20s named find, I assume in her 20s, named Daisy Castro, who played violin, playing modern tunes, but with a feel that reminded me of Stefan Grappelli, whom I love. And we just saw all different kinds, Latin jazz, all different kinds of things. And it was just a tremendous weekend of music, and the weather was nice, and we're on the ocean, and couldn't be better than that. So with that, let's get started. This episode really comes out of two features. One is who the guests are today. And second, and more striking, 5 or 6 times in the last 2 weeks, I've heard a question from investors or observers, never hepatologists, about, well, gee, we're reporting all these clinical trial results now, but given the history, how many of those are ever going to come through? So I said, beats me. You know, my last natural science course was high school biology, as I tell people from time to time. But we're fortunate to have with us people who are significant authors on 2 of the best graveyard of NASH drug papers I've seen yet. One that Jörn participated in 2020 and one that Sven co-authored in 2023. So I thought what we would do was start by asking Sven to run through the state of the graveyard in 2023 and some of the things that he and Mazen Noureddin and Alexander Krag found when they did their paper. And we'll ask questions and then have Jörn comment in the context of what we knew in 2020. And then I've got a couple of questions about what all that means going forward. With that, Sven, my friend, the floor is yours.
Of course, meanwhile, the situation has changed quite substantially.
Yeah, we have a drug now, right?
Because we achieved the FDA approval for the first drug, which is really a milestone, I think. The paper we wrote was on the occasion of the reports of the Falcon trials of pegbalfermin, which did not hit the endpoints, although, as for many other drugs, you had some signals of efficacy also there. So we reflected on that in that paper, trying to, just give an idea about what the problems are with all those failed trials that we have seen so far. The main points were, first of all, you have the definition of the endpoints. There's a lot of discussion about histology as such, although I think that is not the main problem because biopsies are read before and after the intervention in the same way. So that's not really the issue because we discuss a lot about inter-observer variability, which is real. But we know from different analyses now that if biopsies before and after are consistently analyzed in the same way, it's just about difference before and after. So that effect of inter-observer variability disappears there. And even if you have machine learning approaches, you end up with virtually the same results. So it's not the biopsy as such. It's more the endpoints that have been defined based on the biopsy that play an important role because they are high barrier endpoints. If you want regression of fibrosis in a score that just goes from 0 to 4 and we focus on F2, F3, that's putting the bar high. If you look at many of the trials and you incorporate into the efficacy in terms of fibrosis, not just the ones that regress fibrosis, but also the ones that do not progress, so the prevention of progression, which is clinically probably very relevant because if they do not evolve to F3 or F4, they are not likely to have liver-related events because in earlier stages, their clinical events on the long run are not so much liver-related. For the F3 and for sure the F4, then it's liver-related events that is important in their outcomes. But in earlier fibrosis stages, it's not those endpoints. It's cardiovascular endpoints mainly and non-hepatic cancers that play a role. So putting really a prerequisite that fibrosis regresses is putting the bar high. And probably those that do not progress are already having a clinically meaningful benefit on the long run. Still to be proven, but that's probably one of the main reasons why many trials fail because we have put the bar too high. The other aspect is, of course, what this has to do with patient heterogeneity, differences in response. One of the problems with these trials has always been because we have to recruit based on the liver biopsy, which is not always very acceptable to patients. We had sometimes very broad inclusion criteria in terms of all the cardiometabolic comorbidities of the disease. We allow HbA1cs up to 8.5 or 9%. There's always the prerequisite, of course, that they had an attempt of lifestyle modification, but still we enter a lot of patients into a trial that are not optimized for all the cardiometabolic comorbidities that this disease associates with. And then because of that, you will end up with a lot of placebo effects. High placebo effect rate is one of the problems we are encountering in those trials. And you also include patients in those trials that are very heterogeneous in that respect. So the heterogeneity of the patient population is another important factor, and the fact that they are not optimized from a metabolic point, a cardiometabolic point of view. And there are two very important reasons, I think, why a lot of potentially efficacious molecules did not hit the endpoints in those clinical trials. Another point is, of course, that it might be that some of the drugs are not efficacious. This is a very complex disease in terms of pathophysiology. We should realize that a lot of this is driven by what happens outside the liver and subsequently has an effect on the liver. And then the second player, of course, is the liver itself and the way the liver is equipped to cope with all the damaging and stressing factors. And so there is for sure an intrahepatic factor in the disease pathophysiology, but a lot is also driven by what happens outside. And if you have drugs that do not tackle both aspects, both the drivers of the disease extrahepatically and also all the mechanisms intrahepatically, probably some of these drugs might have some benefit, but are a little bit too weak to hit those endpoints. So if you have drugs that are not very potent because of the complexity of the pathophysiology and they are just a little bit too narrow in the whole spectrum of mechanisms on the one hand, and you put the bar very high on the other hand, then by definition, the risk of failure is more elevated. And probably we have missed some of the drugs that might have had some benefit, probably not for all the patients, for at least a subgroup of patients.
So, Jörn, I see you nodding.
Sven raised a number of highly important points, and if you go through them one by one, the one you started out with, Sven, was that from a clinician's perspective, maybe the endpoint of regressing fibrosis is too high, and we want to prevent the progression, and this could be an endpoint for a drug too. So stabilizing the patient. Clinically speaking, that's absolutely relevant. I can see how it's difficult in the regulatory context because we struggle to define a group that actually under placebo shows that relevant progression. You then need to select patients that actually do progress to show a stabilization, because if you select a placebo group that has a lot of early cases or not so severe, you might not see that progression in the placebo group. And I think that's critical, and it's very crucial to think about that when you start your trial and actually plan on the inclusion, exclusion, pre-screening criteria. So this is where this stands and falls, I think.
Yeah, I think it's a very important point, Jörn, and I think therefore it's important that probably one of the things in the inclusion criteria, it's not just what I said about all those cardiometabolic comorbidities that are not always optimized. It's probably also that we are not always including the right patients for pharmacological treatment, because this disease is driven by the activity of the disease, of course, translating into fibrosis for sure, but the driver is the activity of the disease. And with the way we define it, and also in terms of having recruitment feasible, sometimes we did not include patients with very active disease, because the more active the disease is, first of all, there is more fibrosis, but there is also more fibrosis progression. And probably we included patients with too low degree of activity of the disease, which has also been shown, by the way, that if you make sub-analysis more focusing on patients with the most active disease, sometimes you get better results, more clear results, and a more clear separation between placebo groups and active treatment.
It, of course, hampers your recruitment because you're more stringent, and that brings you down again on conducting a realistic trial in a good time frame. It's a two-sided sword.
Yeah, absolutely.
The other element, of course, is that if you do select for the more active, we already know that it's pretty difficult to find people who've got the right level of fibrosis and the right level of activity in all the little boxes, and therefore it becomes a more rarefied group in whom the study is being conducted. And that brings us on to probably falls just outside the remit of today's conversation, but actually applicability once the drug is licensed. It is exactly as you say, you've got to squeeze, it's like that old analogy, isn't it, with a hamburger. You want to hold it tight enough so it doesn't fall over you. So you want to squeeze so you get the right population, but not so hard that it all falls down over your clean shirt, and then you end up with a group of patients who actually don't really represent the population in whom you might use the medicine. Mm-hmm. I have to say, it's a really elegantly written editorial, and I think unsurprisingly given the authors. But I think your point about prevention of progression is real, and some of it actually might be where we started from. So the original point of understanding was about, if you like, I always think that this field was slightly hampered by the success of viral hepatitis that happened alongside us at the very beginning. We wanted to get rid of the injury. We had that mentality. We were a bit scared to say, or not a bit scared, but maybe a little overcautious to say, do you know what? I don't need my liver to look pretty. I just need it not to get any worse. And I agree. I think that prevention of progression piece is really important.
Yeah, in the clinical endpoint trials, the regulators now have accepted that preventing progression to cirrhosis is a clinical event. So prevention of progression to cirrhosis is a potential benefit.
So, Will, I like the word applicability. One of the things that came up last week on this podcast when we were actually talking with Michael Charlton about how he's treating patients in his clinic right now, is he made the same comment, which is the definition of efficacy he's going to use is lack of progression, which makes sense. I think I understand why the agencies wouldn't be comfortable with that one, partly because the disease is kind of heterodox, right? I mean, over time, some people will get worse and some people will get better if you do nothing. So it becomes hard to figure out exactly how to grade out stable disease. And one of the episodes I'm looking to do in the next few weeks is about how other diseases and how the way we think about other diseases might have affected the way that regulators think about this. And I always go to oncology at moments like that, where stable disease, simply maintaining stable disease is an okay thing. But usually if you can't regress disease, they're not interested. So that was my main thought about that one. Sven, I was also intrigued by the observation in the paper that having as a criterion improvement of the NASH score with stabilization of fibrosis might not be a good thing either. I think the suggestion was that the NASH score had too many elements in it and they were insufficiently controllable to know exactly what you were getting.
Yeah. The NAFLD activity score, you mean? Yeah, because it's of course a mixture of steatosis and the signs of inflammation and liver cell damage. And of course the steatosis is relevant, but you can have modes of action where the steatosis, which is very dynamic as we all know, if the improvement in NAFLD activity score is mainly driven by reduction in steatosis, it's not necessarily reflective of less activity of the disease. It's still an important one, but probably not by definition reflective of reduction of disease activity in terms of inflammation and damage. And it's mainly those two that drive fibrogenesis, not the steatosis per se. Although, of course, steatosis is also a sign that something is wrong.
I mean, this introduces the concept again that it also depends on the MOA, right? So we have drugs heavily working on the steatosis side through weight loss or even anti-steatotic drugs, and ones where they're a little bit more neutral on the weight side and have a different ability to change the steatosis here. I think they produce different results, and that means if you consider that endpoint and you make it very sensitive to changes in steatosis, you might not see the improvement of fibrosis based on your MOA you might be looking for. So I think that's important to consider, actually the MOA of the drug.
And it's also, I think, interesting to note that Stephen presented at NASH-TAG also an analysis now separating steatosis from ballooning and inflammation in the NASH CRN scoring system and the NAFLD activity score. So the concept is, which has been introduced already a long time ago in the SAF scoring system, separating steatosis from activity and defining activity only by ballooning and lobular inflammation. Now we are getting more data that this separation makes sense and that we should probably look at steatosis and activity then separately.
So, Sven, if we looked at activity separately, what would that imply for the challenges that are widely reported around assessment of ballooning? Do you still handle it the same way? Or at that point, if you put more weight on activity, do you have to do a better job of handling ballooning than this kind of 0-1 where people don't agree particularly?
Yeah, absolutely. But that's the problem we're also struggling with, of course, is that you have very, let's say, rough scoring systems. I mean, if you have activity in the SAF scoring system, it's just a maximum of 4 points. So you have only 5 different scores to give with an equal weight for ballooning and lobular inflammation. If you take it from the NASH CRN, there's 1 point more for lobular inflammation. So it's a little bit more granular. But you're right, the challenge about the correct identification of ballooning and the quantification of both ballooning and lobular inflammation is a challenge. And I think there machine learning approaches that really standardize the analysis of the biopsy can I think really be helpful.
But I wonder whether we are, within liver disease, being, I suppose, detrimented because we do have the only regenerative organ. So we're looking for that regeneration within the disease. No other disease has that opportunity. So it's about that progression. And I think it's been said here in the last few podcasts, and ever since resmetirom got approval, all about exactly what you raised in the article about the progression. That would have changed the outcomes of some of the trials that were failures because they were looking at different endpoints. That lack of progression for me and the patients that I deal with and the people with poor liver health is and has been really key. And I think it was certainly the patient advocates raised it very highly. Wayne Erskine-Ridge, who has cirrhosis, isn't eligible for the medication and would have loved something for non-progression. All of that certainly hit home when I was reading through. And the other one, you were just saying about the optimization of cardiovascular disease and the other diseases when we come to treat. If we look at that conversely, for all of those patients undergoing current metabolic studies in diabetes, cardiovascular cardiovascular disease, they've not been optimized for the liver health because we don't really assess it. Is there an argument to optimize those patients to locate the ones with the poor liver health that actually can prevent the outcomes in their studies? And that we should, now that we have better NITs, both biochemically and non-invasively, really playing to our strengths and optimizing the location of disease in these patients, cardiovascular and metabolic.
Louise, I think that's a really important point because, as Sven said in the opening piece, a lot of these patients aren't necessarily fully optimized. And, you can look geographically and sort of methods of recruitment and how the trials are advertised. But what you find is that a lot of these patients you might consider in the real world, by the time they've got to a hepatologist who may have done a biopsy, one would hope, I don't think it's real yet, but one would hope that a lot of these patients will be on the best possible combination of medicines for their diabetes, et cetera, et cetera. Now, if you conduct the trial in a setting where you only have extremely well-controlled patients, the likelihood of seeing the activity may be less. And that goes back to Jörn's point around the screen failure rate and actually getting the trial recruited and over the line in the first place. On the other hand, what we would really like to know when we're thinking about whether or not the medicine works is not whether it works in a whole load of people who have not been well controlled and actually simpler other medicines might have reduced their risk of disease development in the first place. But actually what we want to know is whether it's going to have a delta over that. And that's where I think first of all, the trials that are currently being more liberal in terms of their inclusion criteria start to sound a lot more attractive in terms of which medicines are allowed. Of course, the challenge then is if your medicine is one of those drugs that is potentially good at optimizing cardiovascular risk, then you're in a sticky situation. If, for example, you have got a GLP-1 receptor agonist within your experimental arm, then it's going to be very challenging to say, well, we're going to allow a whole load of people on a GLP-1 receptor agonist. Some trials are doing that, but then you're really asking about the delta over that. So we're in a very difficult situation where, if you like, normal or placebo has changed during the lifetime of some of the trials that we're talking about. I think that's really important. And, one could take this argument in a couple of ways. I'd like to know what the impact or how much we can tell by looking at the placebo response rate around this factor. Because again, that's very variable. In the trials where the medicines haven't got over the line, you tend to see higher response rates in the placebo arm. And I think that is probably a good bellwether as to who's gone into the trial in the first place. So I think you're absolutely right. Optimizing and getting people over the line in terms of their CVD risk factors may alter the baseline, the protoplasm for the trial drug itself.
First of all, I think that's a great point. Second, we talked with Dean Tai last fall, who told us, and I think they showed this at ASLD, actually, a paper that Stephen Harrison delivered, that when they use some of their models, they tend to find a pretty consistent 33% placebo response rate, which is higher than reported in trials. Now, they also show much higher drug response rates. In the case of the resmetirom data, they were showing the drug response was 55, higher than reported also. But Dean said they can pretty consistently get a 33% response rate for placebo, which says to me that if we understood how to look at the underlying issues, we might get a higher number, but we'd get a consistent number, which would be hugely helpful. And beyond that, well, I like your idea that goes, we might be able to learn from the nature of the placebo group. If in fact they're all on the same other agents, then in fact that might become some kind of index in a way that we haven't quite thought of it so far. At least I haven't heard it spoken of so far. Sven, you started talking something I did. I'm sorry.
No. It was going back to what Louise said about also the relevance of the response rate, I think also but it's getting back a little bit to what we already discussed, that with, for example, with resmetirom, you can say, okay, if you just look at fibrosis regression, you have only 11-12% effect size. But if you take into account those that do not progress, you get an effect size of more than 30%, 33 or 35%. And so it illustrates that if you look at the total picture, that you get better efficacy data. And I think in terms of the cardiovascular mechanisms that Will alluded to, we should not forget that if people are well controlled, for example, with aspirin, with statins and so on the very long run, even those drugs, they will never hit the endpoints that we define for a MASH trial, but they do have a benefit also for the liver in terms of disease progression and going to cirrhosis and cirrhosis decompensation. So we have all interest in making sure that their cardiometabolic risk factors are treated optimally and then tackle also the liver problem.
No, but I totally agree with your point. But I guess what I'm trying to say is that if you've got a patient, like for like, one with poorly controlled diabetes and one with well-controlled diabetes, then the likelihood that that person will end up in a position where their biopsy shows that they've got active NASH and fibrosis is different. And therefore, if you like, their likelihood of trajectory moving forwards in the short time frame of a trial, be it 1 year, 2 year, whatever, they are also going to be on different trajectories. So absolutely agree with you. Definitely those medicines aren't going to touch those endpoints, but it might be that they have created a different, if you like, substrate in which the drug is being tested.
Yeah. No, you're absolutely right.
I mean, the important point, and well, not sure you meant that, is for the clinical trials, of course, we look for stabilized patients, right? So fully controlled, we're going to have that in real world clinics where there is not an optimization maybe, and then the drug effects could be different, potentially better because you have more ability to bring down the glucose resistance. But typically the ones we include in the clinical trials should be stable the best we could get it. And then, they could still go different trajectories, but they would have been stabilized before enrolling.
They'll be stabilized by the parameters that we measure, by their HbA1c. And of course, glycemia alone, as the diabetologists are finding now, and I'm a total amateur on this, but I would say, but when I hear the diabetologists speak, glycemia alone isn't the only marker of outcome. I see what you're both saying. I just wonder that as we have more and more people on SGLT2s and GLP-1s, whether we're going to find that some of the medicines that have maybe under-recruited patients taking those medicines we're going to find a different effect once they're out in real life. Totally agree. We're going to have to wait until we're out in real life before we answer those questions. But I think who's going into the study is really important. And certainly, I mean, you both recruit very heavily into these trials. When we recruit as well, there's a long list of people who we look through who we never really consider even screening them at the pre-screen rate. So they're never measured on the pre-screen fails. So it's really important to consider who is it that we as the investigators are pushing into these trials. Because, there is a selection here that's ongoing.
Yeah, but I think that we also often forget, and not only we, but probably the field also, the purpose of a clinical trial is different than the purpose of routine clinical practice. In the clinical trial, we really want to demonstrate that the drug itself has an efficacy. And when it then comes to using that drug in routine clinical practice, it's not so much a problem that the patients are not exactly the same as the very well stabilized patients in the clinical trial, because the purpose is different. The purpose of the trial is really trying to show the added value of the drug, because it's always a risk to take a drug. So you also need to demonstrate the benefit, and therefore you need more selected and stable patients in the clinical trial. I don't see really the disconnect between the more strict environment of a clinical trial and then the more broad replication in a more heterogeneous population, etc. I don't think personally that the patient population in whom we eventually use the trial need to be strictly the same as in the clinical trial. But of course, they need to have the disease, but it's about demonstrating the efficacy and so justify the use of the drug.
Yeah, no, I completely agree with that. I guess all I'm trying to go back to is, because the original question of today's conversation is, why have the drugs failed? Or are we to be disheartened by the fact that the drugs have failed? And I think sort of being critical of how we've included patients in the trials may be one explanation. That's kind of what you touch on in your paper.
Yeah, and the background therapy is, as you say, is continuously evolving, and it's now different than the trials we did 5 or 10 years ago.
So let's go back a little further than that and see, what did we learn between 2020 and 23 about how to do things better? And what can that tell us about what else we need to learn now? So, Jörn, I'm going to ask you.
I think the world of clinical trials in the indication of fibrotic MASH has clearly changed a lot since then. The first graveyard article I did, it was with Joost Drent, and you mentioned 2020, so it was looking at drugs like Senecrevarok, and the Selonsertib was out recently, and I'm probably forgetting some of which we covered in there. I think we took a totally different approach in terms of pre-screening, selecting the patients, and reading the endpoints. So by now, we spent a lot of work on refining the way that the two endpoints, MASH resolution and fibrosis regressions, are read consensus among pathologists, not based on a single read. And a lot of things have changed since then. The other thing is that in the initial enthusiasm in the field, there was a lot of MOAs rushed into clinical trials that, after consideration and careful consideration of the preclinical data, you would have probably taken a little slower approach and might have avoided some unnecessary trials. But it was a little bit of a gold rush in the early 20s.
I think that's a very important point. I cannot agree more. I think that one of the reasons for failure of some of the trials is indeed the true lack of efficacy, and that's with those mode of actions and preclinical data, but not always very robust phase 2 human data to go further into the large phase 3 trials with 1,000 or 2,000 patients. I absolutely agree.
I think that's right. One thing I will say, though, is at that time, though, we were looking at the readout of trials that had been designed 5 years earlier. So there was a time lag. There was a sort of a necessary time lag that people in other disease areas didn't have to have, because certainly those first trials had quite a high activation energy needed to get going because of some reluctance in the field. There was some nihilism. We only really had the NASH CRN trial design to go on. So there'd been quite a bit of a lead up to delivering those trials. And although looking back, one might say, yes, that we probably should have had a closer look at that phase 2 trial before embarking on the phase 3 or earlier, I think we learned an awful lot. And I think that it's possible that the field would have been criticized for making too many assumptions without going through some of those studies. I mean, for example, knowing about the rate of progression, which we're back to progression now, knowing the rate of progression in the selonsertib trials, the F3 and the F4 trials, knowing those rates of progression in 2,000 patients, I think gave us a real insight into what the trial population, so people with F3, F4 with NAS scores that were elevated, how that goes ahead as an example, I think is really informative. And although I'm sure investors and pharmaceutical companies don't necessarily want to run multi-million phase 3 trials in order to learn the natural history of the disease, we learned an awful lot as investigators, I feel, from those. So failure, of course, because not achieving the primary endpoint, but in terms of us learning and making the subsequent trials better, I think definite takeaways.
Yeah, that's absolutely right, Will. And I think we should also acknowledge the effort of some of these companies to really dig into those data sets of trials that were reported to be negative. But some of them did not stop it there and have put those data sets available to the community with very relevant information that has really helped us and have advanced our understanding of the natural history of the disease. Yeah, you're absolutely right. In that regard, it was not a waste of energy.
They were clearly visionary enough to get us kickstarted and actually at the place where we're at today.
Sven, I think that's right. One of the points about the earlier trials, certainly the Gilead trials that you made in a different context, but I think is really helpful, is that we were looking to purely intrahepatic modes of action to deliver enough efficacy to make things change. And that hasn't worked. The world is a lot different. A friend of mine who's in dental products, of all things, asked me what I thought the impact would be of the Lilly trial, the Zepbound trial.
Tirzepatide.
In sleep apnea, that we're now learning that the combination agent has a significant effect on sleep apnea, something from like 25 interruptions a night down to 5, which is huge. Not surprising. Sleep apnea is closely correlated with obesity. That's a drug that has a lot of effect on obesity. But it does make the point that everything is interconnected in ways that we wouldn't previously have thought. And that the rise in use of GLP-1s and SGLT2s in all settings has several messages to us, some that we've talked about and some about the idea that, to go back to where you were, Sven, that extrahepatic and intrahepatic effects probably are both necessary to generate sufficient benefit.
Yeah, I think so.
The interesting thing there, if we talk about maximizing patients' condition, is sleep apnea is also strongly correlated to fatty liver disease. And I wonder what non-invasive assessments they are doing to rule out the hard enlarged liver underneath. But just as a point, we can rule it into most things where we're looking at cardiometabolic or anything to do with those conditions, but we're not doing it. We're trying to rule everybody else's diseases in or out of hepatology trials and liver trials to make sure that we're trying to deal with the pure, whereas we're not given the same respect in most other conditions. It's very rarely talked about over 5 times the upper limit of normal when the LFTs raise that they're concerned. So maybe we should be arguing for far better awareness of the liver health and the condition underneath, because that's where we're probably going to help diagnose a lot of people that we're missing in other diseases.
Louise, I think you're absolutely right. I do think, though, I want to shout out to Diabetes UK, actually, because last week was the Diabetes UK Professionals Conference. So this is essentially one of the big diabetes meetings in the UK. And a colleague of mine, our research manager, Jill Hood, talked about it, and we decided decided she'd write to Diabetes UK. So she wrote to them and said, why don't we have a session, an academic session, not a sponsored session, on liver disease? And on Friday, Jill and I, which was obviously World Liver Day, Jill and I chaired a session on Friday afternoon at the Diabetes UK conference at Excel, where we have easels sometimes. And obviously it was a Friday afternoon, and I was expecting, the 4 speakers and Jill and I to be the audience and not anybody else to turn up. And I have to say, the room was full. It was packed. There was some proper debate going on. And much more importantly, it was more full than the arena.
Fantastic. That's really exciting.
Yeah, that's a victory.
Yeah.
His reputation preceded him.
That's a personal win, Will.
I mean, everything's a competition, right?
It's a multiple win. It's a win for Will. It's a win for the liver. Right, and you never any battle with the kidney.
Yeah, and it's a win for Diabetes UK, actually. I think they were really visionary to go, like, actually, this needs to be part of the academic programme, and let's just crack on with it.
What were some of the questions out of interest?
And so, the talks were talks about background of MASH. Helen Jarvis spoke about primary care. We talked about acceptability of pathways and some of the emerging drugs. And the questions were about, the things that we have debated. What's the benefit to the patient? Is there an added burden of excess testing? What do we do when we've found these patients? But to have that debate properly and to have it done in a sort of a proper session, I thought was superb, because we were able to expand some of those arguments. And it's only through that conversation, of course, the conversation went on after the session as well. So I think we just need to get ourselves at these meetings. It's a bit of a I wanted to plug Diabetes UK. To your point, yes, we do need to talk to people in other disease areas.
That's encouraging stuff.
Yeah, I like to present it as vicious circles, because it's multi-directional and everything is pushing everything. So it's not just one pushing the other. It's really turning around. And what we need to do in our management is just stop those vicious circles to turn, or make them turn the other way.
I'm picturing a Rube Goldberg machine being thrown into reverse and watching all the different disparate pieces try to rejigger themselves to go in the opposite direction, right? It isn't a simple task to make everything go in reverse. All right. So what does this tell us what does any of this tell us going forward? We have an audience of people who are in investment or in other way, shapes, or form interested in predicting the future. And knowing what we've learned about what doesn't work should be one of the helpful elements in telling us what does work. So
Roger, let's revisit your first question. Is there ever going to be a drug approved after all these failures? And there are 2 great articles we discussed today, one by Sven and colleagues and one that we put up in the earlier days. And I think that the signal is very clear. Even after that article, there's a drug approved, and we have learned a lot as a field, and we are in the position to get more drugs over the finish line. And we'll be, in the end, mixing and matching those drugs. And I'm very convinced that with all that learning curve, and despite all heterogeneity and some inaccuracy in endpoints maybe, the potency of the drugs we're looking at today is clearly set to overcome those hurdles and get through the finish line.
So I think that's an excellent starting point, right? Which is going back to Sven's point number 5, maybe the drugs don't work that well, or some of the drugs that were being evaluated at that point in time didn't work that well. We have drugs right now that appear to work a lot better. Now, that still doesn't mean that we don't lose aldafermin because of one funky cell somewhere in the middle of the design. But yes, clearly we're seeing more promising results with a more promising set of agents. I'll tell you one specific question I've got, which is if the intrahepatic effects are not robust enough to drive a drug to approval, but are real, will there be a point at which we can marry up intrahepatic and extrahepatic effects in combination agents? And do we have the tools to make that work right now?
What might happen, I think, is that with all the drugs coming in terms of the GLP-1 dual triple agonists, will already drastically change the background therapy of our patients. And probably there might be some direct intrahepatic effects. There are anyhow very strong extrahepatic effects that take away a lot of driving force of the disease, not always completely, but a lot of driving force of the disease. So a substantial proportion of patients will improve their liver health along with their cardiometabolic health. So it will probably shift a little bit the strict indication for pure anti-MASH liver-targeted therapy, which I think is good, but we should consider that the background therapy of those patients is rapidly evolving, and that changes also our field. And I can imagine that in those patients with that kind of background therapy, still having clinically relevant liver disease, a more liver-targeted treatment will be even more important than probably it was before, because you have a lot of possibilities now to improve the cardiometabolic milieu. But still, all with resmetirom, it's different because there we have really a liver-targeted therapy with, besides the LDL improvement, it has not so much effect on the cardiometabolic milieu. So it's really an intrahepatic anti-MASH treatment. And I think that might get probably even a stronger indication if the metabolic background therapy is optimized.
Roger, can I just get, I have to say, I agree with what Sven said. The premise of your question actually is really interesting. That's sort of got me pondering. The lack of efficacy in the liver, if I understand you correctly, would therefore be a reason to partner with a drug that had efficacy outside the liver, as some kind of a fig leaf to cover the reduced efficacy that we're seeing. And I think that as a rationale for combination probably is not a particularly strong rationale. I think the concept of combination for its own sake is very firmly established in infectious diseases, where there is selective pressure, or in cancers, where again, you're applying a selective pressure to something that has a very rapid evolutionary risk of becoming resistant. That, I think, is very cogent. Or you are combining two things where really you want a drug that's got a dual effect. The drug has a dual effect, combined agonists at different receptors in a similar pathway. But I think what we're describing is an a priori, either add-on or type of scaled approach to treatment of MASH, in the same way that we might have 2, 3, 4 agents for one's hypertension. But I think that needs to come because each medicine stands on its own feet, not because one medicine can be brought in to cover up for the other. Unless, I guess, what you're saying is you're going to take through a combination as the drug itself. But then I would want to look at that a bit more carefully and ask, why is it that each individual drug isn't alone, and then you're post-marketing trying to combine the two? And I think that, my only personal view, is that as more drugs get to license on their own merits, people will be asking those questions further down the line. If it were the case that no one drug can get over the line, then I think maybe we will end up in that situation. But that's my view on the combination, which is it doesn't need to be the case that it's going to be combination. We might get there, but the reasons and the routes why we get there, I suspect, aren't quite clear just yet.
So, Will, I think you touched at the end on kind of what I was trying to ask, not very articulately at the beginning, which is that it's easy to foresee a world in which two monotherapies become approved as a combination agent. What happens less frequently, but from time to time, infectious disease being, I think, a good example of that, is that you see therapies approved with multiple agents, and then where at least one of them might not stand on its own, but has an augmenting effect on the others. And thinking of how many modes of action are involved in liver disease, in steatotic liver disease, I kind of wonder whether that might turn out to be a case.
The sort of a metabolic equivalent of ritonavir boosting type thing.
Check. That's exactly where I was going.
Ribavirin story.
There you go.
Yep. Might be, but yeah.
It might be. I mean, the other thing, of course, we could talk about is intra versus extrahepatic effect. I mean, increasingly, if you sort of work in the metabolic space, then, we're seeing, even our own labs, Sven and I talked about this before, you look at what's happening at other sites, and actually that has a direct impact on what's happening in the liver. Work from our lab is showing that cells can circulate, probably the same cells that you see in inflamed adipose tissue are also in inflamed liver tissue. So I think you've got the idea that something might work in the liver or in the adipose or elsewhere, I think may be moot, because it may well be that the same agents, the same things that are causing the injury, are circulating around.
Interesting. Who else has questions or comments on this general topic of what we've learned about trials and what effect that will have on drugs going forward?
Because we've not really been doing a very good job of assessing underlying health in all the medications that are out there and have been approved for all sorts of diseases, we might be blindsided with something that's out there that once we start assessing patients more for liver health and non-invasively, because we can do that now, we might discover it. AKA, you've got that recent study with aspirin. Aspirin has been around for an awful long time. It's obviously potentially been reducing liver fat in people for an awful long time, maybe not to the levels that we needed. I think it was the Harvard study that showed that. So we might actually find that we've got drugs out there that are doing relatively good jobs within fatty liver that we've just never assessed, because we've not been looking beyond a blood test realistically. We've not been doing the level of non-invasive therapies and techniques that we've now got access to. How many studies have really used MRI PDFF to do anything other than our studies here in fatty liver, because that's what we we've been looking at. So if we can expand that into other medications, we may find that there are already combinations out there that could be working, not dissimilar to the ones the guys have described. So I think we could be blindsided somewhere along the line. And as we learn more and investigate more and assess people's livers better going forward, potentially, we may well be able to find more of those medications that are already partially working in some. So I'd like to think there's something out there that is helping that we'll discover because we get better at discovering it.
I agree with Louise that we got to be open-minded and even look for some unexpected combinations. I mean, the challenge is, again, we don't have that NIT that will get us definite approval. And the change in MRI-PDFF in that study with aspirin, which is great, it will need to be shown to prove that on a bigger scale and with more patients and different outcomes. So there's always the caveat of that these trials are so complex these days, it makes it challenging, in particular if you have a drug that's giving to a lot of the patients a priori for their cardiovascular risk.
Well, there are also the economics, which is who will spend money to do a low-dose aspirin trial. Governments aren't funding that scale of study anymore, and no manufacturer is going to make any money on it.
The challenge, it means we shouldn't give up.
We shouldn't give up. I mean, I think it's a fascinating result. And that's the case, by the way, where somebody could put low-dose aspirin on top of the therapy, claim that there's a logic for having them as combination, put them in a single pill and see what you could do with it. Back at odd combinations. Before we wrap, one or two more questions. What do you think the implications of all this are going to be for the timing of approval of future drugs? If a drug is good enough, do we know enough to get it to the finish line faster? Are the things we're learning going to get it there slower? Well, it didn't get there before resmetirom, so slower is probably not an option. But do we know things that will get it there faster?
That's a difficult one.
Yeah, I have lots of questions I don't have answers to. That's why I'm asking them.
To predict, I think not on the very short term, I think it will hopefully increase the investment but in terms of changing, I think we still need a lot to learn how to work with NITs, not just in the selection of patients and the diagnosis, but also in monitoring of disease evolution and assessing treatment response. There's still a lot to learn, so we're still waiting a little bit for that, although the data are coming, so that's fascinating, and I think that will probably make a true change in how this is all evaluated and may accelerate the proof. As long as we are not having that, we still have to wait for the clinical outcome trials, and that's already a shift, I think, in the paradigm of clinical trial design over the last 2 years, so that is promising, but I think for approval, probably we will have to wait until we have a better way of working with the NITs and get them also accepted by the regulatory.
Makes sense. Okay, we're at the bottom of the hour, so why don't we go to wrap up? I guess my closing question is, now that we have a drug approved in the U.S. and coming to market in Europe, how will that affect any of the issues around the conduct of trials going forward as you see it in the context with the things that we've talked about in these two papers, or any other context, really?
I mean, as somebody said at the start, this is a game changer. This is it's a milestone. And certainly in the US, people have already started prescribing the medicine. There are people with MASH taking resmetirom. And I think there will be some questions. It would be great to get some colleagues from the US on to see, just how widespread it is. Is everybody accessing it easily? What's actually happening on the ground? There will be a group of people who have the medicine who will want to think twice before being on a placebo-controlled trial. There will be many people who still don't have access to that medicine despite the licensing. And I think that they're going to be a population for whom clinical trials remain an attractive option. I can't see licensing in UK and Europe. It's not going to be immediate. There is a route through the MHRA where stuff that's happened in the US can be brought across in a more rapid way. But for Europe itself, we've got two colleagues who can speak to that. Maybe that's going to be less of an issue. The next step will be reimbursement. So I think there is a bit of a road to travel before people are going to be on these medicines in Europe and in the UK. So it's business as usual for the time being. And at the moment, for a patient with MASH who is at risk of progression, taking part in the clinical trial remains a trial option. And so in terms of the trials ongoing, I don't see that being a major problem yet.
Yeah, and what I also would hope and expect is that the narrative about screening and case finding will now rapidly change, because one of the arguments has always been, we can screen, but you don't have a treatment, which, by the way, in my opinion, was not right, but still it was in the mind of many people. I think that is also something that will rapidly change, and we will hopefully see much more screening and case finding in populations at risk, and more colleagues that are convinced that this is something that is worthwhile looking for. And that will help also the field forward.
That's a very important point. I remember a quote of Harrison on this podcast talking about shiny brochures are being brought to the physician's office. And I think that Sven just voiced that impression that, the perception among fellow physicians, the referral pathways we've been working on, they're going to be used more heavily because we'll get more attention.
To that point, I agree with Will absolutely wholeheartedly in the context that it doesn't change much here in the context of the current trial recruitment or Australia. Australia don't have a lot of their non-invasives on Medicare, for example. They're not refunded, so they don't have the FibroScan in the same way. It's just not routinely available. So clinical trials are going to be the best way to be cared for and done. It was interesting, there was an article published, I think, for World Liver Day that was the use of non-invasive diagnostic tools and metabolic dysfunction-associated steatohepatitis, a qualitative exploration and challenges of the barriers. Now, that was by Jeff Lazarus, Emmanuel Toxidi, and Miru, and a few others, Marsden. But it only looked at the barriers and the challenges from the physician's perspective. Now, I can assure you, if you were talking to any of the providers of NITs into these areas, we have a lot of barriers trying to be able to bring better ways and smarter ways of getting these non-invasive, whether or not it's Siemens delivering, ELF tests and things like that. There are lots of barriers into the system to be able to help develop. And I think, to Sven's point, we have to sort out some of those barriers in the next couple of years while we get access to these medications. We need to be case finding better and smarter. We need to be bringing down the barriers, and we need to be looking at unique and different strategies to do that and updating our pathways, because some of the fears that they brought out were lack of knowledge. Nobody uses the pathways in the same consistent way. So they were the usual perspectives from the physician and the clinician side. But what we need to do is also have that discussion with the providers of these tests, Perspectum for MRI, PDFF, things like that. How do we bring down those barriers from both sides to help the patient and help hepatology and liver care move forward when we get these medications? Because we can do this. We need to look at it from all sides. But yeah, trials in Europe, I think, will stay for the next foreseeable future. And as we try and bring these barriers down and get better care, really.
Okay, so I'm struck by somewhere that Sven started and Will finished up, and how they go together. If we're going to be in a world where patient heterogeneity has been a little bit complicated and we need to understand more about the patients going into our trials, if you look at the U.S. at a point in time where patients with certain kinds of insurance have much easier access to Rezdiffra than others, that will actually skew the U.S. trial population, right? If the idea is everyone who can get it will get it, everyone who can't get it will go into trials, there'll be some certainly socioeconomic skew in who goes into trials at that point. So the patients won't be as heterogeneous, which is maybe a good thing, maybe not. But the ways in which they're more homogeneous will be interesting. If you see more Hispanics, you're likely to see more PNPLA3, for example, or some of the genetic and microbiome predispositions, certain disease concomitancies. And we're going to have to figure out how to take advantage of that, because obviously those aren't the only populations for which those drugs will work, but they may be the easiest ones in which to trial until at least you get a standard of care that isn't placebo. So I think that's it. If anyone has any thoughts on that, that's great. It's just the thing I got listening to Will's comment at the end. Good. We're an hour and 8 in. Thank you very much, everybody, for your time today. And we'll be back next week. I believe we might be talking about Global Fatty Liver Day, but I'll let you know more about that later. Sven, Will, thanks. Everybody say goodbye.
Bye. Great to see you guys.
Bye.
Bye.
Bye. Good to see you guys. Thanks.
And now, here's Roger with this week's question of the week.
As we discussed in today's episode, the kinds of lessons described in the 2020 and 2023 papers, coupled with wise and creative leadership from Stephen Harrison, among others, have created a far more favorable atmosphere for mass drug development. Still, significant challenges remain. What do you consider the one greatest hurdle left to overcome that will improve the percentage of compounds achieving approval? And separately, the one change that might speed time to approval most?
And now for the Season 5, Episode 12, Back-End Report.
Slava Ukraini, glory to Ukraine, and also Am Yisrael Chai, the people of Israel live. As you probably know, by now, President Biden has signed the bills to extend military aid to Ukraine and Israel, and badly needed supplies are on their way to Ukraine already. Separately, it's horrifying to me that a chain of events that started with the slaughter of over 1,200 innocent Israelis and the taking of hundreds of hostages should devolve 6 or 7 months later to a worldwide rash of anti-Jewish behavior. If you're Jewish, like me, be proud of our joint heritage. If you are not, please help us fight prejudice. Heroyam Slava, glory to heroes, and also Am Yisrael Chai, the people of Israel live.
Rest in peace, Stephen Harrison.
It seems callous and trivial to discuss the tsunami business in our back-end report this week. Instead, I want to take a minute to say thank you to our co-founder, Stephen. This podcast would not exist without Stephen Harrison. This is true in several ways. Stephen was the person who got me interested in what was then called NASH in the first place. When I visited and spoke at NASHTAG 2019 at the behest of Yasmin Rahimi, he was one of two people who reached out to me in the week after NASHTAG to ask what kinds of work we could do together and how I could stay engaged. Stephen and Manal Abdulmalik were the two people at NASHTAG 2020 who encouraged me to open my consulting firm, Hepdynamics. Stephen was the person who called me right after the pandemic and asked the question that led to the formation of this podcast in the first place. And Stephen was the co-host throughout our first year and a frequent guest after that. I'll talk about this a bit more when we do our memorial episode, but for now, let me say this. Stephen, thanks for bringing me to this community and this podcast. It was magical to watch you deliver the major Maestro NASH presentations at recent conferences, as each demonstrated that resmetirom was one step closer to becoming Resdifra, and indescribably sad that you will not be here to witness all the ripple effects of the big fat dent you've put into Basel. All our work honors your effort. And that's it for this week. I want to thank our team, Jörn, Louise, Mike, Eric, Steve, as we keep moving forward. And I want to thank listeners for staying with us. The original plan for next week was to preview Global Fatty Liver Day from the Global Liver Institute. We may stick to that schedule and honor Stephen's work, memory, and inimitable style the following week, or honor Stephen next week. We will post and announce our plan by the weekend. Stay safe, Sirfan. Bye-bye now.
Have any questions for the surfers? You can send them to questions@surfingmash.com and we will answer on the podcast or website. Your question might even wind up being the question of the week.